Qui me connaîtraient pas. Prof ici et membre du CerebrUM. Et je suis ici, j'ai le très, très grand plaisir d'être ici pour vous présenter ma formidable collègue Nim Tottenham, qui est professeure titulaire au département de psychologie à l'Université Columbia à New York. Elle est d'ailleurs directrice même de son département depuis quelques années. Et là-bas, elle dirige le laboratoire de neurosciences affectives développementales. Donc, elle s'intéresse beaucoup au lien entre le développement des compétences émotionnelles, la régulation émotionnelle et la maturation cérébrale. Et entre autres, elle s'est beaucoup intéressée à l'effet des expériences précoces comme le stress, les relations parents-enfants et leurs impacts sur le développement du cerveau. Elle a publié plus de 140 articles, des chapitres de livres étant, laissez-moi vous dire, là, dans des revues vraiment d'excellente qualité, que ce soit en développement, en neurosciences ou en neurosciences développementales. Ces travaux sont très, très bien publiés, bien cités. Vous nous excuserez, hein, ça va arriver de temps en temps. Ça disparaît pendant une seconde ou deux, mais ça revient. Fait que il ne faut pas s'en inquiéter. Oui, peut-être ces travaux sont très bien cités. Quand j'ai commencé à lire en neurosciences développementales affectives, j'ai trouvé que c'était difficile de trouver des articles où ces travaux n'étaient pas cités. Son nom est vraiment là, mais partout dans la littérature. Puis d'ailleurs, elle est très, très fréquemment, ce qu'elle fait aujourd'hui, elle fait ça à tout bout de champ, là, elle est très, très fréquemment invitée comme conférencière partout aux États-Unis, mais aussi partout au monde. Et c'est donc sans surprise qu'elle a aussi été la récipiendaire de plein de prix, que ce soit du NIMH, du congrès FLOTS, qui est un super bon congrès en neurosciences développementales, de l'EPA et même de la National Academy of Sciences. Nim, Sonia, es-tu d'accord avec moi? Nim, c'est une rockstar. C'est une superstar. Donc, on est très, très, très chanceux qu'elle ait accepté de venir aujourd'hui. So, welcome, Nim, and over to you.
Thank you. Good afternoon, everybody. It's so good to be here. Thank you for the invitation and the great warmth you've shown me. I apologize for the flickering, but I think it will be okay. So I'm very excited to share with you some of the findings from our laboratory. We ask a very old question in psychology, which many of you are studying as well, which is how do we explain this enduring link between experiences we have and later emotional functioning, which you can appreciate, especially in the case of the human. This is lasting a very long time, years, if not a lifetime. And so there are many pathways we could investigate to understand what's holding these two endpoints together. And the pathway we focus on is brain development. And because we're interested in emotional behaviors like learning, reactivity, and regulation, we focus very much on the development of the connections between the amygdala and the medial prefrontal cortex. Not because these are the only regions that are doing these behaviors, but they're certainly lying at the foundation of these types of behaviors. And the idea is that the early experiences we have may have some outsized influence on the way that this system constructs itself over developmental time, such that the individual differences we see in emotional behaviors later on reflect that construction process. So in our own laboratory, as well as many other labs, if we look at the growth chart, so to speak, of this neurobiology, here we have age on the x-axis. Many studies find that, well, you don't know what they found. Many studies find that there is strong early amygdala reactivity. So here we are starting at around age 4. And that tends to attenuate with increasing age. And in many ways, this makes a lot of sense because the amygdala's job is to understand the relative safety and danger of the world. And when you're tiny and new to this planet, you have a lot of learning to do. So it makes sense that the amygdala is responsive so early. And yet, this strong reactivity is occurring in the relative absence of adult-like connections with medial prefrontal cortex, which we know from both animal and human studies are critical for helping to regulate the overactivity of the amygdala. So very often studies find that the emergence of these adult-like phenotypes occurs sometime during adolescence. So this has made this period, childhood, very interesting to us because the nature of the conversation between amygdala and prefrontal cortex seems fundamentally different than it does at points thereafter. And it raises the question for us, is this pattern somehow giving us a potential biological mechanism that provides us increased plasticity for the way that the system is going to develop? In other words, is this a potential mechanism for a sensitive period for amygdala prefrontal development? Yeah.
Can I have to clarify?
Yeah, of course.
Has anybody looked at whether the fact that the connectivity is not much happening between 3 and 8, but that might be maybe methodological compound because of the way we compute connectivity, the metrics we use. If the reactivity of the amygdala is not where it is later, maybe there's something there in our metrics.
So it's always a good question to ask. In general, I will say across different methodologies, and I'll show you one in a minute, it does seem that based on the human data, this is real, that the connectivity is different and it's not an artifact, for example, of differential goals during childhood. And in rodent studies, it's also been shown through, for example, tracing studies, optogenetic studies, that you get a lack of those top-down adult-like connections during a, post-weaning prepubertal period. So there's increasing accumulating evidence to suggest that this is a real pattern we're seeing.
Right. So not given by maturity of the tissue, but actually
The connections itself. But we wanted to ask this question ourselves with the tools that we have available to us as researchers of human children, because this is based on correlation, right? This is we're not manipulating anything. And if I were studying non-human animals, I might be able to do something more invasive, like I might want to activate one region to see what consequence it has on the other region. Since we couldn't do that, a former graduate student, Dylan G, said, well, what if we could manipulate one of these regions through a behavioral approach and then see what consequence that has on the other target region? So here we showed participants fear faces, which is a standard way to get a nice bold response in the amygdala. But before fear faces, she presented participants with a block of a task that should activate medial prefrontal cortex. And in this case, it's a numbers group. So in brief, your task is to tell me how many words are showing up on the screen. And the answer is in conflict with the word that you see printed there, right? So the correct answer to this first stimulus is 1, even though the written word is 4. So this type of conflict is known to activate prefrontal cortex. So you would get that hard conflict task, and then you would see these fear faces. And the question is, does your amygdala do something different following this activation of the prefrontal cortex? And is that different for different age groups? Sorry, that was a lot of differences. But in brief, I'm just showing you behavioral data here to illustrate a manipulation check that for both children and adolescents, they're responding appropriately to this numbers group task. It's harder to do the harder task. Okay, so when we look in the brain, we see this medial prefrontal region activating similarly across children and adolescents. Now, the critical question for us is, after that activation, what happened? What do we see in the amygdala in response to those fear phases? For adolescents, we see a relative decrease in amygdala reactivity to fear phases once the prefrontal cortex had been activated. And that's consistent with a regulatory relationship between amygdala and PFC in adolescence. And in childhood, we see the opposite, which is the amygdala activity increases following activation of the prefrontal cortex. So for us, this was just another demonstration that there does seem to be something different going on, and this is not a connectivity measure here, in this neurobiology during childhood. So this, we started thinking about how do we start to build a brain then, given these different patterns across age? And it could be, given the hierarchical nature of brain development, that early on, the conversation between amygdala and prefrontal cortex is largely driven by the amygdala. So I often liken the amygdala to the older sister who got there first and then teaches the prefrontal cortex what it's learned about the world. So the idea would be that this conversation over time leads to the instantiation of those top-down, more regulatory connections from the prefrontal cortex, which again, we cannot conclude based on the human neuroimaging data, but several studies in rodents, for example, show there's merit to this idea because the sprouts from the amygdala to medial prefrontal cortex are emerging developmentally earlier than the reciprocal connections from PFC to amygdala. So what we can do with the human data is subject it to modeling techniques. And this was thanks to the work of collaborators, Steve and Catherine Hansen at Rutgers, where we gave them our data and they returned to us these models indicating the relative direction of information flow. And in general, the data are more consistent with, in adolescence, information flow going from prefrontal regions to the amygdala. And in children, the data are more consistent with a model of information going from amygdala to prefrontal cortex. And then we have the awkward tween years where it's unclear what's happening. So this type of thinking, got us to remembering an old quote from Wordsworth that the child is the father of the man. And the idea here is, in terms of neurobiology, that if we start with an adult vantage point, we often think about the cortex as controlling or driving the amygdala. But if we take a developmental vantage point, we might appreciate that the opposite is true, that the amygdala is really sort of setting the developmental foundation for the way that the prefrontal cortex development ensues. Now, all this development, of course, is not happening in a test tube, but it's happening in a very special environment, which is the caregiver, especially when it comes to the human. And up here, I have lots of pictures of different caregiving arrangements. That's to illustrate that the caregiver could be the mother, could be the father, could be the grandparents, the aunties, uncles, could be adoptive parents. And that's an important point because this is a learned relationship. So nobody's born attached to their caregiver. And in some of my talk, I'm going to focus on what's the basis of that learning. The other thing I want to point out is, although many species in the animal kingdom spend time with their caregivers, humans enjoy some outlier status when it comes to how long we spend with our caregivers. So in humans, we're talking on the order of years, decades, depending on the structure of your family. And that is a very interesting design that Mother Nature gave us, because it is metabolically very expensive to raise another human being for a couple of decades. So what's the trade-off? And one argument is that the trade-off we get is this incredible prolonged plasticity of the human brain. So this long childhood affords us kind of a scaffolding of emotional brain development that's coming from our parents. Now, if that prolonged plasticity is something that we get as humans, we have to appreciate that, of course, plasticity is a double-edged sword. On the one hand, this prolonged plasticity may increase the risk for mental illnesses associated with early life stress, for example. You can see here on the x-axis is age and a number of mental illnesses associated with emotion regulation challenges plotted on the y. And their peak clinical age of onset is typically happening sometime between the transition of childhood and adolescence. On the other hand, this incredible plasticity is the basis for our epistemic growth, our ability to gather massive amounts of information from our unique environments and become the sophisticated adults that we all are. Now, during development in humans, there's a very interesting balance between our great drive for epistemic growth and the associated risk-taking and exploration that accompanies that growth, and our great need for safety and security early in life. So, for example, this child here is able to explore his environment precisely because of the presence of this species-expected stimulus of his caregiver. So, in other words, the parent enables efficacious exploration for the child by being a source of security. And so it's for this reason that people like Alan Strofe have reminded us that the caregiving relationship is an asset to the child and that it affords them the luxury of exploration and risk-taking. Keep checking these wires just in case something magical happens. Okay, so in maturity, we have intrapersonal means of making sure our safety and security is maintained while we explore. And this is largely being taken care of by connections between amygdala and medial prefrontal cortex. And the premise of our work is that the caregiving relationship plays a central role in establishing this neurobiology. You just made it worse. Thank you. This is actually an experiment.
Really?
It's change detection. Okay, so so these ideas are not necessarily new. Many of them are baked into the foundation of Bowlby's attachment theory. So this quote I find so compelling from some of his writing. In the countryside in springtime, there's no more familiar sight than mother animals with young. So familiar are these sights, and so much do we take it for granted that lamb and ewe will remain together, that the questions are rarely asked. What causes these animals to remain in each other's company? Why do we follow our parents? It's kind of a magical behavior, right? Some animals don't do that. Sea turtles don't do that, but we will follow our parents. And the second question is, what function is fulfilled by their doing so? And today I'm going to focus on fear and safety learning. When we think about fear learning or avoidance learning and safety learning or affiliative approach related for learning, we often think about them as being on opposite ends of a spectrum. However, developmental psychologists have often pointed out that actually they're much more intimately related than we might think. So understanding affiliation learning requires an understanding of fear learning in the context of development. So in some early work on affiliative learning and fear learning, it was identified that there is a specific timeline for the onset of these two forms of learning. Specifically, there's a maturational delay such that affiliative learning precedes fear learning during development. So, for example, in this picture here, you can see that a puppy who's exposed to a human handler during a critical period for affiliative learning, occurring here, will exhibit affiliation for that caregiver. But if you wait just a little bit longer, let the puppy get older and then expose them to the human handler, this same puppy would instead exhibit fear. So this is demonstrating that maturational lag. And the argument is you want this lag in an altricial species, meaning any species that depends on caregiver attachment, because fear learning is going to naturally interfere with the affiliations we have to make with our caregivers. So you kind of want to keep this quiet for a while so you can get this set up. And this affiliative learning is so robust that it will occur in the presence of shock. In fact, it will increase in the presence of shock. And so it's for this reason that Bowlby wrote, early affiliative learning seems to defy the principles of reinforcement learning, that there is this obligatory preference that young animals like humans and puppies will show. So in other words, altricial young will affiliate regardless of valence. So more recently, this effect has been demonstrated by Regina Sullivan's group. If you're unfamiliar, in brief, she pairs a peppermint odor, a neutral odor, with a foot shock. And following this pairing, she'll place the animals in a Y maze with that peppermint odor on one arm and a control on the other. Very young animals, rats, will show this bizarre preference for that peppermint odor, even though they do not like the shock. She knows that. They're showing this approach-related behavior. Whereas if you wait a little bit longer, that animal would instead show an avoidance like you and I would. And it's been argued that this is happening because of that maturational delay. This is happening because in the young animals, you want them to demonstrate a preference for a learned cue, regardless of the valence. They will show this preference, of course, if the peppermint odor had been paired with something rewarding. But what's amazing to me, anyway, is the fact that they're also preferring the shock paired stimulus. And this has been argued to mimic the learning that occurs for the young animal to recognize who its attachment figure is. Now, if you let this animal grow up a little bit, it turns out that the presence of the mother, now that the mother is, I'm using mother because these are rats, but the presence of the caregiver will later, sorry, this is to establish attachment, will later facilitate preference learning for other new odors. In other words, this now becomes another odor associated with my mother, right? Like my mother used to wear Chanel No. 5, now she went to Yves Saint Laurent. So I know that that's mom, right? That's the idea. And this is what that looks like. These are now those older animals who would normally avoid the peppermint odor as you and I would. If you fear condition them now in the presence of their mother, she reinstantiates that infantile phenotype. They're now showing a preference for the peppermint odor paired with a shock if it had been learned in the mother's presence. And the argument here, if this is feeling not intuitive to you, is that for the young animal, their only survival mechanism is to attach. They can't fight and they can't flee. The only thing that they can do is attach. So the young brain is designed perfectly to do that because it's been beneficial for our survival. And mechanistically, what Sullivan's group has shown is this is largely due to the mother's effect on the amygdala. So here's rat age on the x-axis. There's this period postnatally when the functioning of the amygdala is dependent on mother's presence. So when the mother is in the nest, she is keeping cortisol low, which is keeping dopamine in the amygdala low, which is keeping the amygdala relatively quiet during fear conditioning. When the amygdala is quiet during fear conditioning at this point in development, there's competitive learning systems that can kick in, piriform cortex, olfactory bulb, that will mediate the learning and produce a preference. If you take the mother out of the nest, she's away, those processes reverse, and the amygdala is free to participate in fear learning, so you get the avoidance behavior that you and I would typically show. So these data are a nice reminder that the developmental state of the young altricial brain is not deficient. it's perfectly designed to coordinate with parental input. And these processes are important for attachment learning. It helps us do two things at once. One, maintain proximity to parent-related cues, and two, potentially explore frightening stimuli. So we were very eager to ask if humans show this effect of the parent on fear learning behaviors. So we brought in preschool-age children and we fear conditioned them to this, I'm calling it fear condition, to this blue square. We didn't shock them. We instead used a horrible noise like nails down a chalkboard, and they also find it terrible. So children would learn the association of the blue square with the horrible noise. That was the CS plus, and the triangle was paired with nothing. They learn this association either alone or with their parent. The parent's not doing anything. The parent's just physically present. Okay. After the association, the parents went away and we made a human Y maze. So children entered the Y maze and the CS plus and CS minus were placed on different doors, and we asked them enter a door and to get a prize. And we showed them it was the same bucket of prizes behind both doors. We just wanted to know which door they preferred. And we found that when children fear conditioned alone, they did more or less what you and I would do. They avoided that nasty blue square. But if they fear conditioned in the presence of their parent, they started to show that behavior that the rat pups did. They were now showing a relative preference for the blue square. We were curious to know if some of the mediating process involved differences in attention. So we used a dot probe task. If you're not familiar with it, you put both competing stimuli on the screen. You don't have to do anything to those stimuli. On the next screen, you see an asterisk on one side or the other, and your job is to press a button to tell me where the asterisk was. And the idea is that the speed with which I respond to the asterisk will indicate my attention on the previous slide. And here we found that when children conditioned alone, they showed a relative avoidance of attention. They averted their attention away from that blue square. But when they conditioned with their parents, they now showed a relative attentional bias towards that blue square. And this is a within subjects design. So these are the same children whose behavior, attention is changing as a function of that manipulation. So to ask if the amygdala is, the human amygdala during childhood is responding to parental cues differentially, we presented children with a very simple task here. We presented them with 30 seconds of a picture of their parent, and then 30 seconds of a picture of somebody else's parent, so a stranger. So the simple block design showed us that during childhood, we were getting a relative decrease in amygdala reactivity to the parent stimuli. So at least at face value, it seems that we're interpreting this as some form of parental buffering. Now, in this block design, we weren't fear conditioning children, so we felt a little bit weird about calling it parental buffering. But in a recent paper from our lab where fear conditioning occurred in the scanner, parents were either in the scanner with children holding their hands or they were in the other room. We see this effect as well, that the parent's presence is bringing down amygdala reactivity during childhood. And it's momentarily, phasically, the parent stimulus is phasically strengthening the functional connectivity between amygdala and medial prefrontal cortex. So one idea this generates is that the routine day in, day out interactions we have with our parents are in some ways laying down the tracks of what is to become our future mature emotion regulation neurobiology. So everything that I've said so far requires that the parent is calm and regulated. So in the human studies, the parents are calm and regulated. They're just sitting there. In the rodent studies, the mother is anesthetized, so she's very regulated. And we know at the same time from a large clinical literature, as well as rodent literature, that as effective as a calm parent is in regulating children's fear and stress, a dysregulated parent is really effective in potentiating children's fear and stress. So to model that here, we did another fear conditioning study, but we didn't fear condition the children, we fear conditioned the parents. So for example, here's me at the top getting fear conditioned to this pink circle. I'm wearing headphones. Every time I see the pink circle, I hear that screech and I flinch. So we record that and then we show it to my daughter. And then she sees a second video of somebody else's parent getting fear conditioned to the diamond. And then later we test her on these two shapes here. And we find that after viewing those two videos, her amygdala seems to care more about the shape that their parent witnessed relative, was fear conditioned to relative to the stranger. And in terms of behavior across time, children learn better to dislike. Higher scores means learn better to dislike. Well, they learned to dislike both stimuli. They knew that they were both bad relative to a CS minus, but they learned better about the stimulus that their parent fear conditioned to. So taking these types of findings together, if we consider age on the x-axis here, this first decade of life may be a time when parents are especially able to modulate activity of amygdalin-related neurobiology. Perhaps parents have some sort of privileged access to these deep limbic learning regions. And we have some ideas about the neurohormonal cascades that might be affected by parent availability or by parent distress. But the idea is that what's happening during this time is foundational for producing the individual differences that we see in emotion regulation behaviors emerging during adolescence and beyond. So with this framework, it allows us to now start asking different questions about what happens if during this first decade of life, you're exposed to caregiving related early adversities, which for the rest of this talk, I'll use the acronym CREEP. And I'm specifically using this acronym, which you may find annoying. But anyway, the reason why I'm using it is because I want to emphasize that it's caregiving related. So there are unfortunately many types of adversities that young children can be exposed to. But our premise is that when it comes to emotion-related neurobiology, it's those experiences that are most targeting the integrity of the parent-child relationship that are going to have the biggest impact. So when I say CREEs, I'm referring to these types of experiences. So extreme experiences. So abandonment or permanent separation from parents, abuse and neglect, multiple caregiving switches, and or institutional care. So like orphanage care. And in brief, many studies have shown findings like this. If we look in broad brushstroke terms, in terms of internalizing problems like anxiety and depression, or externalizing problems like conduct or potentially attention problems, we see that a history of CREEs is more likely associated with increased problems. However, I put the individual dots here to show you the widespread, right? There's a lot of variation in here. And some of our questions are aimed at asking, well, what differentiates these two children here? So when this is some early work, we just asked the basic question, are we seeing differences in amygdala prefrontal activity as a function of these early experiences? And the answer is yes, we see amygdala hyperactivity in response to fear phases. And we see altered connectivity between amygdala and medial prefrontal cortex. At first, we might have thought that this would be some sort of delay here following early life stress, that the system's not working the way that it should because of exposure to some toxic stress. But actually what we saw was the opposite. We saw a pattern that more closely resembled adult-like patterns of connectivity following early CREEs exposure. So the idea is if this is age here on the x-axis, and the typical age that we see amygdala prefrontal connectivity emerging is around the beginning of adolescence. Given the hierarchical nature of brain development, early amygdala hyperactivity in response to these CREs may lead to an earlier instantiation through an activity-based means of these adult-like connections with prefrontal cortex, because maybe they serve an adaptive benefit for the individual today. If so, we know with adaptations that they often mean trade-off. And so this might lead us to predict a trade-off such that in a future time point, we may see some cost associated with that earlier instantiation. So for example, if indeed there is a reduction, an acceleration, which I'm saying hypothetically at this point based on our data of this development, does that correspond to a reduction in that developmental plasticity during childhood? So we see this in the human data, but there are also many findings, and this is a short list here, from rodent studies consistent with that idea that you see earlier adult-like phenotypes, and work in this department shows that you don't necessarily even need to go to more extreme exposures to see how the caregiving environment is playing with the timing of this neurobiology. So if it's true that we get a reduction in that plasticity, then it generates the hypothesis that this accelerated development may place limits on that parental buffering that we see of amygdala. So we repeated that task of 30 seconds of your parent's picture, 30 seconds of somebody else's. These are the data I showed you before, following a typical caregiving experience of decreased buffered amygdala. Following CREs, we don't see that same decrease. If anything, we see a slight potentiation. So at first, this is suggesting at the group level, we are not seeing that same buffering in childhood by the parent. I should mention the children that we work with largely have experienced adoption following these early CREs and are living with a different family than they were born into. But these are really large error bars. And so what that suggests is a lot of variability. Some of these individuals, even following CREs, will show some buffering and some won't. So we interrogated those individual differences longitudinally to ask, does it matter? Does that phenotype matter in the long run for something like your anxiety? So we split the participants into those who showed buffering of the amygdala at time 1 and those who didn't. And if you pay attention to this side of the screen, I'm plotting here anxiety in both of those groups in following CREs. And you'll see they're not different from each other, although they're both higher than the non-exposed comparison group. But when we invite those participants back 2 years later, those who showed amygdala buffering at time 1 were the ones who had significant decreases in anxiety, whereas those who maintained higher levels of anxiety did not show that buffering at time 1. So what's the difference between those two groups, those who buffered and those who didn't? The variable that mattered for us was the child's attachment. So what I'm plotting here is the child's reported felt security with their caregiver, with their current caregiver. And those who showed buffering at time 1 were reporting higher levels of attachment. So I think these are correlational data. We don't know about directionality. It starts to hint at, in our data, at the power of later caregiving experiences being important in the wake of earlier CREs. There's great work from other labs, including one I was able to collaborate with, Mary Dozier's group, that does use an RCT, and we've been able to collect neuroimaging data from that group, suggesting that the direction truly is from the caregiving relationship to child outcome. Okay, so make sure I have enough time here. So, okay, at this point, we started getting very interested in the phenomenon in the literature, which is the amygdala and medial prefrontal cortex don't just come up in our small subfield. It comes up in a lot of subfields in neuroscience. And so, for example, in the adult literature, in the emotion regulation literature, it seems to be altered following early caregiving adversity. We've shown it's modulated by parental cues during development, and so on, and the list grows. And there was a very nice conceptual paper by Roy and colleagues suggesting maybe this neurobiology does something much broader. And maybe this neurobiology, at least in the human, is really representing schematized emotional knowledge. And this was an attractive idea to us to think about, you know, when we took all the language I've used in this talk so far has been very verb-like this region is processing, this region is doing something. But I really like this paper because it changed my thinking to start wondering about, well, what is the content knowledge that's represented in this neurobiology? And so one idea is that maybe one developmental story is that this neurobiology is really sensitive to caregiving cues early in life. And that's important because it's going to give rise to individual differences in emotion regulation by way of knowledge, emotional knowledge becoming schematized across time. So let me use an example from And so the hypothesis is engagement of emotion regulation neurobiology that we see in adulthood kind of a reinstantiation of early caregiving experiences? Not explicit reinstantiation, but some really kind of primordial knowledge that we have. And if so, is this occurring through a process of emotional learning and neural consolidation over the course of development? So using an example from cognitive neuroscience, there are many findings showing that our establishment of semantic knowledge occurs from by way of the hippocampus encoding episodic information. And over time and repetition, that episodic information becomes abstracted and represented in midline structures like medial prefrontal cortex and posterior ACC, you know what I'm saying, posterior cortex. So for example, you know, the first time, so I actually need my slides. Okay, so let's say the first time I met Annie, okay? So I thought, oh, Annie had very good insights and was very kind to that student, right? So that's the first time. And then over time and repetition of meeting Annie, I lose the details of those original, that original information, but I abstract and represent in these midline structures now the semanticized knowledge that Annie is brilliant and compassionate, right? So that's the idea from cognitive science. So what if there's a parallel process in the affective domain? Because the amygdala is kind of does episodic learning too, albeit a very different type of episodic encoding. But what if the amygdala learns and over time and repetition of the same type of learning, that information can also become abstracted and represented in these midline regions? So our hypothesis here is that the development of amygdala cortical function reflects a non-declarative affective memory structure. So schematized, I'll call it attachment knowledge. So in this example here, imagine an infant having a need and having a need met or not met, whatever the case may be. And you can bind that information perhaps at the level of the amygdala, so that over time and repetition and development, you now build the knowledge when I have needs, they are met. And maybe that's the content knowledge that's stored in these midline regions. So we're just now, I'm telling you about a kind of new headspace our lab is in, and I want to share with you in my remaining time some of the work that's diving into this idea. So the data that I'll share with you come from this larger sample here. I have their characteristics listed, and this sample is enriched for these pre's listed here. So to first ask, is there any merit in this idea at all? We got together with a great assistant professor in the department, Chris Baldessano, who has really, I think, created a really interesting paradigm where he's able to look at how the brain represents schematized knowledge, but he's looking at adults and not interested in emotion at all. And I said, Chris, can we use this approach to start looking at these attachment schemas, or I'm calling them attachment schemas. So we showed participants movies, 3-minute movie clips that had an attachment narrative to it. So some of you may remember this movie with Shirley Temple, The Little Princess. Probably most of you don't. And that's good because our participants also shouldn't have seen this movie or been too familiar. But the 3-minute clip had this important arc. The characters are together, then they're separated, and then there's a reunion. And half the participants saw that movie, and the other half watched Homeward Bound, which has a lot of perceptual differences, but also has this arc of characters being together, separated, and then reunited. And we just collected resting state during this movie watching, and we performed an inter-subject functional connectivity analysis, which is a nice approach because it gets rid of irrelevant connectivity and homes in more on connectivity that's associated with patterns identified in these movies. And what we found was we just did something really simple. We seeded the amygdala and we asked the whole brain, who else is talking to the amygdala while you're watching this movie? And we found that for children who reported stronger sense of attachment security, the amygdala was talking more with the medial prefrontal cortex. This was just an initial hint to us that there's merit in this idea. It certainly requires a lot more. And Anna Benucci, who's a current graduate student in the lab, wanted to dig into this a little bit more deeply. So she wanted to manipulate stimuli and ask the brain, who cares about these stimuli? So we adapted stimuli from an old paper by Susan Johnson. I think it's wonderful stimuli she used with infants during a looking time paradigm. And I'll show you what this looks like. Okay, so in the scanner, children saw these types of stimuli and these types of stimuli. So one, the oval's leaving, one, the oval returning to the smaller oval. And our idea was that if you're walking into this task with the affective knowledge that my needs will be met, then you may have a prediction of return. You may have a prediction that this oval was supposed to return. So this oval not returning creates a violation of expectation. And in contrast, if you walk in with knowledge, my needs will not always be met, then I may have a prediction of leave. And so when I see the oval return, is that going to be a violation? And we interrogate these brain regions to see if they're responding accordingly. So this is a work in progress, but what Anna's done is something called an inter-subject spatial pattern correlation, which in brief, what this analysis gives you is a metric of neural unison. So she could look at patterns that the brain is making in response to these stimuli and see who else are you in unison with in all the subjects. And then ask, is that unison measure associated with having a common early caregiving experience? So what she's found is in response to the final event of the oval leaving, a history of crease was associated with greater unison. So if I have a history of crease and you have a history of crease, our brains are going to be more similar in its activity to the leaving stimulus. But that's not true in those without a history of crease. That unison that you and I are exhibiting is going to be associated with less activity in those brain regions, meaning more activity for the comparison group for this leaving oval. And the opposite was true
when the oval returned. So if the oval returned, then if I didn't have a history of Crease and you didn't have a history of Crease, our brains are going to be more in sync with each other. And that translates into more activity in the Crease group for the returning oval. So it's, we have a lot more work to do here, but it could be that what we're starting to see here is some prediction error that's emerging as a function of these movies. We also looked at a control area that we think is, that we know has been associated with social processing, so the angular gyrus, which does not show that double dissociation. Okay, so I think this is my last set of things I want to talk about. Anna also wanted to know, well, is there a way behaviorally that we can actually ask participants what is in that content knowledge? And so we paired up with Simona Getty, who's an expert in children's memory development. We adapted one of her tasks to be an attachment-based false memory task. So I'll show you how this works. Children were shown these pictorial narratives. These are the characteristics of the participants. So children might see something like this. A mom and daughter are playing together. They continue playing together. At some point, the child makes a bid, an emotional bid to the mother, and then there's this ambiguous ending. So children saw, I think, 27 of these types of stories, and later on, we gave them a memory test. Now, 24 narratives. So unbeknownst to the participants, there's something's missing here. You didn't see what actually led you to this ambiguous ending. So to make sense of this narrative, you have to insert your existing knowledge into this story to make it make sense. So we asked children, did you see this picture? Did you see this one? Did you see this one? And so on. So in truth, children saw almost none of these pictures here. These are all false. They never saw these pictures in any of the narratives. But the two that we really care about most of all are these two. So in one, which we're calling the causal lore, this is a picture that would be consistent with a secure script, right? The mother picks up the child after making the emotional bid. The insecure lore, the mother is scolding the little girl for crying, okay? So the question is, do you endorse these, even though both of them are false? Do you have a bias in which you endorse? So in response to the causal lore that's consistent with secure, we saw no difference in false endorsements between those with and without a Crease history. But for the insecure lore, a Crease history was associated with more false endorsements of saying, yes, I saw this picture, even though they did not. So this gives us some insight into the schematized knowledge that children have. But we also saw a moderation by current attachment security, such that in orange, these are those with a Crease history. The more secure they report feeling today, the less they're likely to make that error. So what this suggests is both the early as well as the later experiences matter here. And I'll just end with where Anna's taking this, because I think it's really exciting. She's saying, well, if we do start thinking about this schematized affective knowledge as being able to make predictions, because that is what attachment is, it's making predictions, then can we take advantage of predictive coding to start to quantify some of these representations? So I'll just reference you to this paper that just came out. In brief, the idea is that if this is developmental time here, early in life, we start off with quite large prediction errors, but there's learning that occurs heavily in a bottom-up fashion that over time transitions to these more cortical regions as that prediction error decreases as you gather information and as your prediction precision increases. And this model then gives room for thinking about how we change our predictions based on brand new information, how we might accommodate these attachment schemas in the face of drastically changing social emotional environments. So with that, we see human amygdala cortical circuitry develops very slowly, but there seems to be some value to this slow pace. We get a late transition to adult-like phenotypes, which gives us long sensitive periods for environmental influence on brain development. The parent-child relationship is a product of learning, and this learning may be potentially unique early in life, with young showing an obligatory preference. And parents can be seen as being primary social regulators of cortical limbic development, perhaps having privileged access. And the developing circuitry may be part of the neural basis of our primary interpersonal affective schemas, with our working hypothesis that the neurobiology underlying emotion regulation is constructed by our caregiving experiences and represents schematized interpersonal affective memory. So with that, I will acknowledge funding, members of the lab, collaborators, the families and individuals, and thank you for your attention.
Told you she was good.
One of the most impressive parts of your talk was your
Own PhD in handling the congratulations already because of that. So we have time for questions, comments.
It was super interesting. Thank you very much. I was wondering, have you tried to study this trajectory of like the impact of parent buffering on children, since it's like a super long period?
Like, when does it stop?
Not necessarily when does it stop, but like how impactful it is at one point, and is there like lower and higher points during that period?
So in our hands so far, we generally find the first decade of life is more impactful on subsequent behaviors. And other groups have found something similar. We haven't had the sample size to be able to look more closely, which I think is what you're getting at. But my prediction would be that the earlier you go, the more impactful you're going to see it. In fact, one data slide that I didn't have time to show tells us something about timing. And this is work that Anna Benucci, who I mentioned, has done. So in brief, she used random forests with a number of variables. And what I'm showing you here is predictions in a never before seen sample based on a model developed from the data. And where am I going? So here she's looking at P factor, the general psychopathology score. And what she found was the timing variables that mattered certainly was the duration, how long were you living in that type of experience. But the second most important was experiences in infancy, so 0 to 17. So, so that speaks, and many studies find something similar, but that speaks to the question you're asking, which is, well, this is kind of a negative event, but we would expect the opposite in more nurturing, supportive experiences.
Thank you so much, Nim. Each time I hear you, I have a new grant application to write because I have so many ideas, but I'm going to take a big jump. Could it be possible that your knowledge schema prediction, the information, you gain from early on, could explain a very, very weird finding from the group of Danise at the Royal College in London? So what they did, we know that early adversity is an interesting, important predictor of mental health disorders later. And we thought that, this is a typical thing. But, most of the studies use questionnaires to assess early adversity, so where you're exposed, etc. So they use subjective reports of early adversity. They don't really have data on objective adversity. But Danise and his group have a longitudinal study that they've done, and they compare the predictive value of subjective versus objective, because they had access to, database where children, sexual abuse were reported, etc. and they look whether such subjective when or objective would be, correlated with later on. First, they showed no correlation between subjective and objective. So it's not because you say that you were exposed necessarily. You may not have said it before. But the best predictor of mental health disorder related to early adversity was subjective reports. And for many years, I was, trying to figure out why. Could it be possible that, the mechanism that you're showing now could modify cognitive processing and lead to these weird data that he's reporting?
Yeah, it's not a big jump, and I think it's a great question, and I really love those data too. The way that I read those findings is really related to this idea of, I didn't use this word in this talk, but meaning making. So I wonder if the subjective reports, so now you're many years out, right? So there's going to be individual differences. Some people who experience A report A, some people who experience A don't report A, right? So what's the meaning that you make of your life experiences by the time you're an adult? And I think it has a lot to do with these centralized representations, right? So these are schema. This is how you make sense of the world when you don't have the full picture. So I think that's one part of it. And the second part of it is, why aren't subjective and objective correlated with each other? There's so many reasons, right? So there's sort of the unfortunate reasons, which is just like people forget or people make stuff up, or right. So there's that possibility, but it's also the methods by which you collect prospective versus retrospective. So the prospective, this is from Kathy Whittam's data set, the prospective are coming from court reports, court findings, right? So what gets reported in court findings? It's going to be largely sexual abuse, physical abuse, and physical neglect. It is unlikely to be emotional maltreatment because you don't go to court for that. So in fact, when Anna looked at the different types of experiences and their predictive value for P factor, the number one experience is emotional abuse, right? And then the second one is switching caregivers, so permanent separation from your caregiver. So these two emotional indices are the best predictors over and above some of these other. That's not to say these aren't important, but in terms of their predictive value, I think the fact that they're not going to get picked up by prospective objective measures is very important. The other thing I'll say here is we did this, I'll show you the slide as it was meant to be presented. It was going to look like this. And we wanted to know, okay, we're going to use machine learning to tell us which of these different experiences is the most important. And you can see that there's some variation with the emotional experiences being at the top. However, those effects are dwarfed by just asking, did you experience any crease? And that means that, yes, the types of things that children experience are important, but not nearly as important as knowing that the integrity of the parent-child relationship was significantly hit. So, so I think it's a great question you're asking, and I think there's a lot of meat there to chew on in terms of thinking about what is the value, right? When I first saw those findings, I was like, that's so depressing. But actually, the more I thought about it, I was like, wait, that's actually very interesting, which was their interpretation too. Yeah.
Thanks for the grant application.
Am I a co-PI?
Yeah, I wanted to ask a question about when you're talking about the variance and the distribution of mental health outcomes or whatever of people who have had CREs. I like that you went a little bit in the direction of like later caregiving situations, because I feel like when I hear that pointed to, people immediately jump to like, what are the constitutional factors? Or like, what's the genetic factors that determine like someone's resilience? Like, for example, Alice Moore had this idea of like an enlightened witness, so like basically a secondary or tertiary caregiver that could, you know, kind of make the difference or like steer a young person's life in one direction or another. So I was wondering what's your perspective on that kind of like nature nurture debate, that specific debate?
Yeah.
But also like how do we even begin to study it? Because it seems like something really hard to determine.
Yeah. Great question. So when it comes to, well, first of all, when it comes to the human in general, I think there's going to be a large bias towards nurture. Not exclusively. I mean, obviously we live in a dynamic system, but I think nurture is going to play a big role here with emotions because it kind of makes sense, right? By definition, we're a species that is adaptable. That's the whole point of being a human. You know, that story of where humans emerged, it was like drought and then it was flood and there was drought and flood. And that's where humans came from. So by definition, we're adaptable. And the reason we're adaptable is because we have childhood. Child is the thing that makes us adaptable. So, so anyway, but in terms of data, Michelle Achterberg, who comes from Evelina Krona's lab in the Netherlands, had this really nice twin study with neuroimaging and showed that the connections between the amygdala and medial prefrontal cortex are best explained by environmental influences, right? So from there, where was I going to go with that? So yes, I think it's the environment. Oh, and so your question is so important, which is I focus a lot on early experiences, but we do have the opportunity, for example, it's not experimental, but quasi-experimental by looking at families who have experienced adoption, right? There's a big change in caregiving. So here we have the heat maps for all of these adverse CREs, and this is the P factor, and then whatever mental health outcome you want to look at. But if we also look at current caregiving, right? So now their children have been adopted. You can see that there's this different additional in the opposite direction effect of the current caregiving environment. And that I think is really important point that you're bringing up, that it's both early and ongoing have to be considered to get a full picture, because there's a lot of variation in the current caregiving situation that could mitigate some of these early effects.
And a teacher or a coach or
That's right. That's right. That's right. If your parent is unable to provide something, then another mentor, right? The mentor effect is really important. Thank you.
Could you tell us a bit about, I never heard about inter-subject functional connectivity.
Yes.
So what are some say, advantages of the approach?
So the basic method is we would seed my amygdala, and then we would ask your brain and everyone else's brain about connectivity. And then you start to get some commonalities across people. And the benefit of doing this is it helps get rid of some of the extraneous connectivities that one might see, and helps you really home in on the stimulus specific patterns that emerge during a movie, for example.
Il y a pas de questions tout de suite. Je vous invite à rester avec nous. Il y a un cocktail comme d'habitude, donc venez prendre un verre. Ce que je vous ai pas dit au début, c'est que Nim, c'est l'une des personnes les plus, je suis sûre qu'ils sont bien d'accord, les plus fondamentalement gentilles de tout le monde académique. Fait qu'hésitez surtout pas à venir la voir. Je suis sûre que ça va lui faire plaisir. Nim, that was a beautiful talk. Thank you so much.